the 28th Samuel Eiduson Student Lectureship. It's my honor to both welcome you and to welcome Lyle Kingsbury, who will give today's lecture. Lyle will be introduced by his mentor, Weizhe, in just a couple of minutes. So the Samuel Eiduson Student Lectureship is being named for Samuel Eiduson and is given since 1993 by a graduate student in the neurosciences at UCLA who's being considered by a committee to be having a research story of particular interest to the broader community. And you will see that that really is true for Lyle, in that he really has done an enormous body of work that is ready for the big screen, I think. Samuel Eiduson was himself a neuroendocrinologist who's worked at UCLA for a long time. In the context of graduate education, he was the one who in 1972 started the interdepartmental program for neurosciences for graduate students and was actually the first chair of the program. He served until 1985. When I came to UCLA, I still remember during the Eiduson lectureship that he was always sitting in the front and interacting with the speakers and actually asking very pointed questions of the speaker. So it's in his memory and in honor of not just the winner or the lecturer for this year, for the 28th lectureship that we're congregating here. It's actually also to honor the work of all the other graduate students, many of which have done absolutely amazing work in this past year too. So without further ado, I'll give over the virtual microphone to Weizhe.
Thank you, Felix. So I'm Weizhe Hang. I'm proud to introduce Lyle Kingsbury, this year's recipient of the annual Samuel Eiduson Award. So Lyle grew up in New York and graduated from Hunter College with bachelor's degree in 2015. And in the same year, he joined the Neuroscience graduate program at UCLA. Lyle is the first graduate student in my lab. It has not yet been 4 years since he joined my lab, but he has made absolutely fantastic accomplishments. Lyle is extremely motivated and excels, especially in difficult and challenging situations. I'll mention a really brief story. So he first contacted me in 2016 and asked if he could do a rotation in my lab. And it turned out that Lyle was going to join another lab, but in the last minute, there was some funding issue that he couldn't join that lab. So I told him he could do a rotation, but my lab space was not ready yet. Fortunately, my colleague Payman Goshani was extremely generous and offered that we could use his lab space. So I told Lyle, if you wanted to do a rotation with me, you have to do it in Payman's lab. That was the start of his time in my lab. I would say that it was quite a difficult situation for him, but it was immediately clear to me that he is not someone who sits around waiting for opportunities. He's the one who creates opportunities for himself. Despite difficulties, he did amazing rotation with very little guidance from me. I hope his story can offer some inspirations for the first and second year graduate students who are experiencing difficulties starting your work in this crazy year of 2020. Lyle is also a critical thinker and a careful observer. It was him himself, not me, who made some of the most important observations that he's going to present in today's talk. It challenges many, if not all, suggestions I made to him, which is absolutely the right thing to do. And when he accepts the suggestions, he always asks me what he could do so that he could think of that idea in the first place next time. It was him who learned and brought new methods and ideas into the lab, many of which I don't even know or heard of. It has been a pure joy for me to work with him and to witness the growth of such an extraordinary young scientist. I'll leave it to him to describe his own exciting findings, but his work really transforms the way we understand the neural processes that underlie social behavior. And he has done so much that he only has time to present a small fraction of his work. Over the last 4 years, an ARCS Foundation Fellowship, a neural microcircuit training grant, and an NIH F31 predoctoral fellowship. He has published 3 first author papers in Cell, Neuron, and Transient Neurosciences. This award, the annual Samuel Eiduson Award, is a special recognition of his amazing achievements and his contribution to the neuroscience field. As part of the award, Lyle is going to present a lecture today on his work, The Social Brain in Action, Neurodynamics Within and Across Interacting Animals. Congratulations, Lyle.
Oh, thank you so much for the kind and humbling introduction. Let me share, let's see, gotta make it smooth. Okay, we're good, we can see the screen. I need to see some feedback from the people. Okay, there we go, great. Okay, well, yeah, again, thank you so much. And thank you all for being here. It's really a pleasure and an honor to have the opportunity to share some of the work that I've spent the last couple of years engaged with under the guidance of Dr. Hong. In the lab, we are very interested in this question of what is going on in the brain when we interact with other individuals and how different systems in the brain help to coordinate social interactions. There are a number of reasons we're interested in these questions. Probably first and foremost, is that we live in a world that is in a lot of ways socially constructed. We spend a lot of time engaged in interactions with our friends, our family, our colleagues, and these day-to-day interactions really ultimately shape a great deal of our life experience. This fact of being social is something that we share with other animals in the animal kingdom, including rodents and mammals that are sort of like us and other creatures that are very much not like us. So this is a feature that is sort of ubiquitous throughout the animal kingdom and shared across many species. And because social interactions are so important to our day-to-day life, we can also recognize that negative social experience can be extremely painful and distressful, or lack of social experience, I should say, and can really contribute to our overall quality of life, and in extreme cases can lead to the development of conditions such as anxiety, depression. I think that this point is particularly salient in the era of COVID, when this global pandemic has in some sense caused an experiment in social isolation for all of us, which is, does not feel good. So, for a number of reasons, we really think that this question of how the brain controls social interactions is a really critical goal of neuroscience from both a basic science and more clinical perspectives. Despite the importance of this larger goal, understanding how the brain coordinates interactions has really been a challenge for neuroscientists. And in part, this is because of the unique complexity that animals are faced with when making social decisions. So let me just tell you kind of what I mean by that. If we think about a situation where an individual is engaged in a static environment, like I'm sort of just sitting in my living room by myself, no friends around, we can roughly think about my decision-making process as sort of mapping sensory inputs to behavioral outputs, and this is maybe modulated by different internal state processes. The environment is not particularly reactive to me, and so it's for the most part fairly predictable. By contrast, in a social interaction, the environment that I'm faced with becomes another agent with their own goals, their own intentions, their own ideas, and by virtue of this, the behaviors that each individual engages in become sort of the immediate input space to the other agent, right? And this sort of forms a reciprocal feedback loop, such that the decision process of both animals co-evolve, and their decision process becomes a lot more complex and contingent on the immediate social context. So because of this complexity, we tend to take multiple different complementary approaches in the lab to understanding the social brain. And today I want to tell you about two projects that have really come at this question from two sort of distinct perspectives. The first perspective that I'll talk about is what I'll call this computational system perspective. So this is sort of the more standard cognitive neuroscience view that we're all fairly familiar with, where we're sort of thinking about the brain as an operator that transforms sensory inputs into behavioral outputs. And the kinds of questions you may be interested in here with respect to social behavior are, where are social cues encoded in the brain? How are they encoded? And how is that information really mechanistically translated into behavioral choice? We can also take another perspective. We sort of zoom out a little bit and think about a social interaction as itself a system, a coupled system of interactors. And this kind of perspective may yield insight into how an interaction actually evolves by virtue of the coupling of interactions between individuals. And we may also want to ask questions about how the neural dynamics underlying those decision processes are interrelated and how that has to do with the interaction itself. So I'm going to come back to that perspective later, and hopefully by the end of the talk, I will have convinced you that both of these views are useful. But for the first part of the talk, I'm going to take a deeper dive in this first perspective and sort of think about how social cues are encoded in the brain and how that really relates to the behavioral choices an animal makes. So zooming in here on this idea of a sensory motor transformation in the social context, we can think of the brain as taking information and sort of processing it in a number of stages. Initially, the brain is faced with sensory inputs, such as olfactory cues and pheromones, maybe vocalizations from other animals. And this is sort of the raw data, right, that the animal gets. These data may be processed further downstream by other circuits that extract features such as sex identity of another individual, maybe male or female, the novelty or social status of that individual. Those variables are then potentially integrated with internal state variables like motivational anxiety state or social context variables. And finally, in this simplified view, these variables are then read out to produce a behavioral output, such as maybe aggressive behavior or a mating behavior that is sort of adapted to the circumstance. So it's probably clear from this kind of picture that in the lab, we work on mice as a model system. And in the mouse, this step of recognizing sex identity is really of critical importance for dictating how the animal behaves in sort of proper behavioral selection. And in fact, at the neural level, the encoding of sex in the brain, recognition of sex identity, is linked to animals' behavior. So for example, animals that have that can discriminate sex cues more readily at the neural level tend to show heightened aggressive behavior or mating behavior, suggesting that this internal representation of sex is really causally related to those behavioral choices. Still, though, most of the observations in the field in this vein are largely correlative, and so we don't yet have evidence that internal representations of sex actually play a causal role in behavior. If we zoom out a little bit more, we can appreciate another knowledge gap that exists here. So historically, in the social behavior field, people have focused mostly on the role of subcortical regions, including the amygdala and hypothalamus, in order to understand how social sensory cues are processed. And there's good reason for this. These brain regions are situated fairly close to the sort of sensory side of things, and they do play an important role in sensory transduction. However, it's long been known that cortical regions, including the medial prefrontal cortex, play an important role in regulating social behavior and in a number of cases causally control social behavioral states. And so this raises a question. If those brain regions, cortical regions, play a role in regulating behavior, then what is their role in representing these kind of important social variables that are known to play a role in controlling those behaviors, right? How are regions like MPFC involved in sex representation, and how is that related to behavior? So we were very intrigued by this question of cortical involvement in sex representations and social behavior, and especially for a brain region like MPFC that is known to be involved in social behavior. And we decided to delineate a couple questions that we thought were worth exploring. So first, do MPFC neurons encode this important variable of sex identity? And if so, does an encoding of sex in the cortex in MPFC relate to social behavior in any meaningful way? Before going on, I want to just actually take a small break and ask, can anybody see my cursor? And is that, you can see the cursor? Okay, great. I didn't know if I was pointing at anything before. Okay, so to address these questions, we wanted to be able to record from populations of MPSC neurons during naturalistic social behavior. And I'm sure many in this audience are familiar with this technique of microendoscope calcium imaging, which allows us to record calcium dynamics, neural activity dynamics from populations of cells through these miniaturized head-mounted microscopes that are mounted on animals' head. We really owe a enormous debt of gratitude to the collection of people at UCLA, including Alcino Silva, Payman Golshani, Balcock, and Daniel Aharoni, who together with others have developed a beautiful open source platform for miniscope imaging that makes this technology very affordable and accessible for new labs like our own and many other labs in the world now. With their support, we were able to adapt this technology to record from MPFC neurons in animals during natural social interactions. And we wanted to ask first, just how do these neurons respond during natural investigation to male and female conspecifics, to address this question of whether MPFC encodes this variable of sex. To do this, we used this technique by mounting these scopes on animals' heads. And while we were recording from MPFC neurons, we presented subject animals with novel male and female conspecifics, allowed them to just freely investigate those animals. And each animal saw about 8 novel male and female conspecifics. And we interleave those investigation trials with presentations of a novel toy object as a control. The first question that we wanted to address was just, do single neurons seem to respond to these social investigation events? And do they encode sex identity? Using an ROC curve analysis, we identified subgroups of cells that seem to respond either non-specifically to social interaction or preferentially to either male or female interaction. And over the whole population of MPFC neurons, about 22% of these cells seem to respond in such a manner. And we wanted to ask really, how do these single neuron responses look at the population level? Are they stable and reliable enough to be able to form a readout at the population level that maybe a downstream circuit could use to tell what the sex identity of the animal is that the subject is investigating? And in order to address that question, we constructed linear decoders to decode the sex of novel investigation events based on population activity. These decoders, as you can see, performed greater than chance levels and fairly reliably, suggesting that these cells formed a stable population code that could be used to read out sex identity. Things got a little bit more interesting when we zoomed in and started to look at really the way these sex cues are encoded in MPFC neurons. So in male animals, we found that female investigation events elicited a stronger response at the single neuron level. So there were more cells that seemed to be tuned to female cues. And consistent with this, the population response in MPFC was stronger, was higher amplitude when male animals investigated females compared to males, suggesting that really the encoding of female cues in the MPFC of male animals is stronger, sort of more salient. And that's represented here in this cartoon showing the population response as a population vector. And you can see the response associated with female investigation is longer, suggesting that that's a stronger response. By contrast, in female animals, we saw no such asymmetry. So there were comparable fractions of male and female responsive neurons. And at the population level, there were similar sort of responses. And so this really suggests that there is a dimorphism here across male and female animals, such that male animals encode social cues sort of asymmetrically with a bias towards stronger encoding of females. And this was not something we saw in the female animals. So we wanted to see if we could link this in some way to behavior, if this had anything to do with behavior. That was one of our goals. And as it turns out, male animals are known to show a preference towards behavioral interaction with females. This sex preference is actually something that is observed in many social species, including humans, which we may have some anecdotal experience with. And in our data, we saw exactly the same thing. So we saw that male animals tended to prefer interaction with females. However, what you can also appreciate here is that there's actually quite a big spread of individual animal sex preferences. So even though at the group level, there's this bias towards female interaction behaviorally, some animals showed a strong female preference and others even showed a strong male preference. And so this raised the question of whether this encoding asymmetry that we observed in the male animal's brain may be related to this kind of behavioral bias that we see in animals. And so we really wanted to know in individual animals, does this coding asymmetry relate to behavioral bias? And the way that we decided to investigate that was to leverage this natural variability that we observed and look at how these sex cues are encoded in different subgroups of animals that were either strongly female preferring or strongly male preferring. When we did this and looked at female preferring animals, and we looked at how these cells were responding to male and female investigation, we found, sort of consistent with before, that female investigation elicited a stronger population response, suggesting that that these cues are encoded more strongly in female preferring animals. And this is consistent with the sort of group level behavioral bias that males showed towards female investigation. Interestingly, though, when we looked at this subpopulation of male preferring animals, we saw the opposite result. So male preferring animals show a stronger activation in response to male animals. And what this suggests is a model where the neural representation of sex in the MPFC is related to sex preference by setting a sort of preference state. So animals that have a stronger representation of female are biased towards female investigation behaviorally, and vice versa for male investigation. Now, this is, of course, still correlative, right? And so what we really wanted to do to test this theory was to manipulate these subpopulations of male and female encoding cells and ask what happens when we manipulate the cells in terms of the animal's behavioral bias towards male or female. In order to address that question, we first wanted to get sort of a better handle on how these variables are encoded in MPFC neurons among different subpopulations of cells. And so we recorded using different molecular markers, specifically from excitatory or inhibitory neuron subpopulations. And as it turned out, these sex-specific neurons were actually pretty widely distributed across excitatory and inhibitory neuron subtypes, really suggesting that these sex cues are distributed broadly across MPFC populations. And what this means is that if we want to really manipulate these cells and see what they do functionally, we need to get a handle on the specific groups of cells that are defined based on their coding identity, that is their native activation profile. And so in order to do that, we could use activity-dependent labeling. And specifically, in our case, we use this synthetic promoter ESER to drive expression of channel rhodopsin, specifically in subgroups of cells that were active during different kinds of social experiences. And in essence, what this lets us do is sort of tag these male and female encoding neurons and activate them optogenetically in a environment where we can get a really straightforward behavioral readout of male and female sex preference. So we performed these experiments, and when we activated female encoding cells, what we found behaviorally was that there was an increase in female-directed interaction, but only female-directed interaction. Interaction directed towards the male animal did not change at all. And so this really meant that activation of female cells biases the animal towards female-directed interaction, increasing its sex preference. By contrast, when we activated these male encoding neurons, which is a separate subpopulation of cells, we saw no such difference in female interaction, but actually saw an increase in male-directed interaction. So the exact opposite of what we saw when we activated the female cells, thus shifting the animal's behavioral bias towards male interaction. And what these manipulation studies really show is that the encoding of sex cues in the brain that we identified using our miniscope imaging is not really just representational, but is related to a functional role of this circuit in setting a preference state and really controlling an individual animal's preference towards interaction with male or female conspecifics. So we set out to look into the cortical role of sex representations and how that may be related to social behavior. And what we found was that MPFC neurons do indeed encode sex identity. We found that using miniscope imaging. male animals showed a stronger encoding of female cues compared to male cues. And this was not just representational, but actually reflective of a circuit mechanism at MPFC that sets a behavioral state by controlling an animal's individual preference for male versus female interaction. So this really, with the study, what we've really done is established a novel role for cortex in encoding these sex cues, something that was largely thought to be mostly subcortical. And we've also, beyond that, found a causal role for sex representations in the brain. So this sort of closes the loop there of the sensory motor transformation in a nice way. So maybe before moving on to the second part of the talk, it's a good time to take questions. I think, Weizhe, I think you're muted.
Yes, so there are a couple questions, clarification questions that maybe you can really quickly address. So Felix has a question about whether in the sex preference experiment, the animals are familiar with each other, whether the target animal is a familiar animal, and if it's not, whether let's say you put the male target animal in, could that male animal become an intruder and say elicit aggression from the subject animal? And on the female side, if you present a female animal, could that female animal, is that female animal in the same stage of the estrous cycle, and or did you use that random or always use the same estrous cycle, and how that could potentially affect behavior? I mean, just maybe really briefly.
Sure, briefly, yeah. So the animals that we use so just to clarify the question, are we talking about the investigation assay or this two-chamber preference assay? Was that?
Maybe you can comment on both.
Sure. So in both cases, all of the intruder animals are novel animals. The question of whether activation of these neurons produces any sort of further social behavior, such as like aggressive or mating behavior, is a really, really important one that we, I think, want to address. It's not something that we addressed in this study, but I think that it would be really, really interesting to see if that increase in what looks like sort of motivational state to engage with males or females then translates to heightened propensity to engage in those kinds of sex-directed behaviors. In terms of the estrous cycle question, we didn't look specifically at whether estrous cycle affects the responses of these animals to at least the behavioral level. But I think that's also an interesting point. I would sort of speculate that that female animals that are in estrus may be more interesting to the males, and that may in fact be one of the important context variables that participates in shifting this preference state sort of dynamically, right? If you're a male and you're in a context with a female in estrus, you may want to shift the motivational state of the male towards more investigation of females. So I think those are both really, really interesting directions to explore that we just didn't get to quite yet.
Yeah, I think a related question is whether when you look at the response to, let's say, a male or female, is that towards one individual, like they test one repeatedly, where you are doing that towards multiple animals? Yeah,
so in this first assay that I discussed back here, this social investigation assay, we presented the subject animal with 8 completely novel intruder animals. So the responses that we see in these neurons that seem to respond on most trials to each of these novel conspecifics suggest that those cells are really encoding sex identity, a general feature across those animals and not an individual animal. Although I think it's an interesting question. There are some other studies that have looked at encoding of single animal identity in MPFC. That may also be something this brain region does. But these cells seem to respond generally to that variable of sex identity. Okay, so maybe I'll move on to the second part, and then we can maybe come back to some other questions at the end, cycle through all these. Okay, great. So until now, I've mostly been talking about the role of a single animal's brain in sort of transducing these sensory signals into behavior. But of course, a social interaction involves more than one individual, right? So now I'm going to sort of zoom out and come back to this perspective that I alluded to earlier, this coupled systems perspective. We're really considering the whole system of animals and as one interactive system, one coupled system. And intuitively, if one is trying to understand such a system, we want to know not only what each part is sort of doing, but also how each part interacts with each other part, right? That is sort of a higher level understanding of what a system is doing. Usually, though, in social neuroscience, we do only focus on one brain at a time, right? Because it's sort of a lot easier to do that. And of course, that's kind of the mode that we were in when I discussed the project that I was just talking about. But we sort of got to thinking, what more can we really learn about a social interaction if we sort of step back and take this multi-brain perspective and really examine how neural dynamics may be related to one another as the behaviors of these animals in the interaction co-evolve. So using these miniaturized microscopes, we made a lot of use of this technology in these studies. We were able to record from two animals at the same time to really sort of address this question of how do the neural dynamics look sort of at this higher level or considering all of the pieces. We extracted MPFC neuron activity from two animals at the same time while they interacted in naturalistic interaction. And we really wanted to just see what was happening when animals were allowed to do whatever they wanted, right? Completely unconstrained setting. So initially, we just put two animals in a box together after outfitting them with these microscopes, and we sort of just let them do whatever they want to do for about 12 minutes or 15 minutes. And to just get an initial sense of what the MPFC neural dynamics are doing, we took an average over all of the cells within each animal, and we plotted these against each other. And we were sort of struck initially to find that these time series signals seem to look fairly highly correlated. And remember, this is, sort of unexpected because these animals are not connected to one another, right? These are completely physically disconnected systems. So why is it that these signals are correlated? We got very interested in this question, sort of digging into what the reasons were. And initially, we quantified this using just a correlation coefficient, fairly simple measure. We found that these signals were correlated across animals around 0.3, which was substantially higher than chance level and also higher than this between pair control, where we analyzed the correlation of signals that were derived from separate animals that were each engaged in interactions, but just not with one another, right, from separate sessions. So this really suggests that this may have something to do with the fact that animals are in an interaction. But there may be some caveats here, right? We're natural skeptics. And one important caveat is that these animals share an environment, right? And so they may experience the same kind of ambient sensory cues like lighting or noise. And so we really wanted to know whether this phenomenon was largely driven by just sort of the fact of being in a shared environment. To look into that, we did a fairly simple experiment where we just introduced a barrier between the two animals. So in this case, they're still just in the same environment. They're still able to kind of do whatever they want. They just can't interact, right? That's kind of all we're disrupting. And when we do that, we found that this interbrain correlation was significantly reduced, suggesting that it really does depend on ongoing directed interaction between a pair of animals. So we were a little more confident that this was sort of something real we were looking at, but still sort of surprised and scratching our heads about what it might mean. It turns out that there have been some studies in the human world, human neuroimaging world, that have looked at a very familiar phenomenon, very similar phenomenon. So there have been a number of studies that have found using fMRI and EEG that there are these correlated activity patterns across human subjects when they're engaged in different kinds of social interactions, games, shared cooperative tasks, things like this. And really interestingly, the degree of correlation often in many studies predicts interesting variables, behavioral variables about the interaction, such as shared attentional states, shared goal states, and also relational variables like romantic status and even dominant status. Until we did this study, it had basically been unknown whether a phenomenon like this existed in animals that were not primates. So with our observation of this in mice, we now think that this is something that extends across potentially multiple social species, which is really interesting. Still, though, we're sort of left the question of why this happens, right? And unfortunately, these human imaging studies, amazing as they are, they can't really get at the question of biological underpinnings because they are limited in terms of their cellular and circuit level resolution and their recording techniques. And so we became very interested in this question of how this interbrain correlation signal is really related to activity in underlying neural components, that is individual neurons. And this was something that we felt we were very well poised to address because we have this nice model system. And because of the technique we're using, we have access to single cell activity. So this really sets up the guiding questions for this second part of the talk. We were very interested in knowing how does interbrain synchrony arise from activity in individual neurons? What is the underlying basis of this phenomenon? And how does it relate to the social interaction? Can we learn anything deeper about how the interaction is evolving by looking at this sort of higher level shared signal? So to drill down into this first question, we initially just set up sort of two broad hypotheses that we wanted to delineate. First, one idea is that these signals are correlated because just broadly the activity in MPFC is sort of co-modulated, and this is a dimension of activity that may involve sort of all the neurons uniformly, right? Sort of just one underlying signal that's co-varying. Another possibility, though, is that these region-level signals are correlated across animals because of the contributions of specific subpopulations of neurons that may play an important role in social behavioral decision-making or encode different kinds of social variables. And so to sort of delineate these, we noticed that what we really needed to do was to understand what individual cells are doing, right? And unfortunately, the task that I described before, this open arena environment, has a lot of advantages, but one disadvantage is that the behavioral repertoire of the animals is very highly variable. Animals tend to show very different kinds of behaviors. And so it becomes quite difficult to really get a reliable understanding of what potential neural correlates there are in the brain of different kinds of behavioral variables at play. So to really address this question, we adopted a different kind of behavioral assay called the tube test. This assay is typically used to study social dominance interactions, social competition. And in this assay, two animals are put in a one-dimensional linear tube, and they're allowed to sort of just push each other back and forth. And what typically happens is that the dominant animal in this pair will push his opponent out to the other end of the tube, and the subordinate animal will sort of just retreat from the interaction. We were mostly interested in this assay because of the way that it really constrains the behavioral repertoire of the animals, gives us a much more tight control over what kinds of behaviors they can engage in. Basically, they can engage in this approach behavior, and then they can push and retreat from the conflict, and that's essentially it. So with this assay, we first wanted to validate that we still saw this correlated neural activity across animals in this different kind of social context. We repeated the same kind of experimental design where we simultaneously recorded animals. We found that these signals were correlated around 0.3 and higher than chance levels. And also when we introduced a barrier, we found what we saw before, which was that this interbrain correlation was substantially reduced just by virtue of disrupting the interactive component of this experience. So armed with this knowledge, we could next turn to the question of what single cells are doing. And using a similar approach that I described before, we were able to identify single cells, groups of neurons that encoded specific behaviors that the animal was engaged in. So here are examples of cells that encode push behavior, retreat behavior, and approach behavior specifically. Altogether, these groups of cells constituted about 35% of the neurons that we recorded. And we can now sort of get to our core question, which was, are neurons that encode social information preferentially involved in contributing to this interbrain synchrony signal? To address that, we performed what is basically an analytical legion experiment where we just removed these subsets of behavior encoding neurons from the population analytically and asked how correlated the mean level signals were. When we did that, we found that removal of behavior encoding cells substantially reduced this interbrain correlation signal. And this was true even if we removed them from only one of the two animals, yet removing an equivalent number of non-behavior encoding neurons didn't really do anything to the signal. So this really suggests that these behavior encoding neurons are preferentially involved, supporting our second model of pattern subpopulation activity. Now, to some degree, this is sort of expected because in an interaction, sort of by its very nature, the behaviors of the animals are somewhat coupled to one another. And so if you have neurons that respond to individual behaviors, you expect that to some degree they are going to be correlated at the mean level. And that is, in fact, probably what is happening here. However, what you also can notice is that when you remove these neurons, there's still a large deal of this interbrain synchrony signal sort of remaining, suggesting that these behavior encoding neurons do not actually fully explain what's going on. And there may be some other information that's encoded in this brain region that is contributing to this signal. What we found when we looked more deeply into what these cells were doing was that there were other neurons, separate but somewhat overlapping with those behavior encoding cells that responded specifically to the behavior of the opponent animal. So here are examples of cells that respond to opponent push behavior, opponent approach, and opponent retreat. And you can see that they don't respond to the subject's own behavior. And together with the population of subject encoding neurons, these behavior encoding neurons constituted, 50% of the population. So half the cells in MPFC encode some sort of social information about the behavior of one animal or the other. When we remove these opponent encoding cells, and play the same game as we did before, we find basically the same result, that these also contribute to interbrain synchronization. And critically, when we remove both populations of subject and opponent cells, we see a sort of aggregate additive response, such that when we remove both of these, we see a very reduced interbrain synchronization down below 0.1. So this result really suggests that it's the aggregate activity of these neurons that encode subject and opponent behavior that give rise to this phenomenon. And this really makes a lot of sense, right? Because both animals have these subpopulations that encode subject and opponent behavior. Whenever anybody does anything, there's going to be a modulation in MPFC of both animals, right? So this, we think, is why the signals at the mean level are correlated across the animals. So armed with this explanation of a sort of underlying neural basis, we next wanted to sort of dive a little bit more deeply into what these cells are encoding, right? They encode subject and opponent behavior, but is there anything interesting about how those variables are encoded, potentially related to the roles that these animals are taking in the interaction? And you'll recall from what I described before with the tube test, that we can measure in the tube test relative dominance. So what we did was we identified the dominant animal in each pair, and we analyzed the single neuron activity in neurons derived from dominant or subordinate animals using generalized linear models. So here we expressed the activity of single neurons as a function of all the different behavioral variables that we measured. And what we did when we analyzed, what we saw when we analyzed the coefficients fit to these models was that there was a really intriguing asymmetry in how these behavior variables were encoded across dominant and subordinate animals. So what I'm showing here is that subject behavior is encoded more strongly in dominant animals, and opponent behavior is encoded more strongly in subordinate animals. What this means is that the behavior of the dominant animal is really more strongly encoded in both animals, right? Behavior of the dominant drives single cell activity across both animals. And that observation actually makes a really cool prediction about interbrain synchronization at the mean level. What it suggests is that if both animals' brains are influenced more strongly by dominant animal behavior, that interbrain synchronization will be higher during behavior of the dominant animal. And this is exactly what we found when we looked into this. So interbrain synchronization was higher within each pair on average during behavior of the dominant animal, suggesting that the dominants really drive the interaction both at the behavioral level and at the neural level. So this was really cool. And we were very satisfied with being able to understand sort of an underlying neural basis for this phenomenon, but we were still left with the question of what this might mean and whether it's sort of relevant to understanding anything about the system. So we wanted to really understand more specifically how this synchrony signal is related to the interaction of the animals behaviorally. And what we did to address that was to look at the relationship directly between neural synchrony and a social behavior interaction. What this graph is showing here is the probability of behavior of one animal, animal B, as a function of time centered around the onset of behavior of the other animal. So this is really a metric of behavioral reciprocity. And you can see that this there's an uptick in behavioral reciprocity following behavior of one animal, which is natural. This makes sense because the animals are in interaction. But what's super cool here is that if you look at these probability curves and you split them up based on the degree of neural synchrony that precedes behavior, that we see this nice relationship where when neural synchrony is higher, this predicts a higher degree of reciprocity following that. And over the whole span of correlation values for the neural correlation, this significantly predicts the degree of behavior interaction of the animals. So this really suggests that this synchrony signal is informative of a shared behavioral state across the animals that predicts the degree to which they exhibit sort of reciprocal interactive behavior. We can go even a little bit further than this. So if interbrain synchronization predicts sort of microscale social interactions, and these behavioral relationships between animals are formed as a result of sort of all of these different interactions over time, then we may hypothesize that neural synchrony predicts the development of social relationships that are a function of those interactions, right? And this is indeed exactly what we see. If we look at interbrain synchronization in just the beginning of the experiment, that significantly predicts the difference in dominant status across the two animals within that pair, suggesting that that when two pairs have a higher dominant status, they are differential, I should say, they are more synchronized with one another. And this is consistent with the idea that more dominant animals are better at driving the interaction, both at the neural and behavioral levels. Critically, this relationship depended on the inclusion of subject and opponent encoding neurons and was essentially non-existent when we replaced those with non-coding cells, really highlighting the fact that this neural synchrony signal and its behavioral meaning really are based on the underlying components and what variables those cells encode. So this is sort of now a complete picture that we painted. Interbrain correlations arise from individual neural components that encode social information, social variables of both animals. And by virtue of that, and the fact that these signals are encoded asymmetrically across the two animals, this synchrony signal also predicts the degree of interaction between the two and their development of dominance relationship between them. So just to sort of summarize this section, I first showed you that interbrain synchronization arises during social interaction in mice. This is the first time that this has been identified in animals, giving us a kind of a nice foothold into potential further explanation of biological underpinnings. Neural synchrony emerges from cells that encode self and other behavior. So this is the sort of basis for it. And it also predicts social interactions and relationships. So in the future, we're really very interested in sort of digging deeper into whether neural synchrony emerges because of certain genetically or anatomically defined circuits in the brain, looking into regional heterogeneity in interbrain synchronization, looking into whether we can use this higher level of analysis to understand anything about deficits in social interaction between individuals, and also in expanding the framework to potentially understand the relationship between neurodynamics across individuals and larger scale group collective behavior. So I hope that in the last 40 minutes or so, I've convinced you that in order to understand social behavior and social interactions, we really benefit greatly from taking multiple different perspectives. This computational system perspective can tell us a lot about underlying mechanisms of how sensory cues in a social context are processed and how that relates to behavior selection. And this zoomed out view can potentially tell us information about how the interaction actually unfolds by considering the interrelationships of neural dynamics across the participating individuals. And so with that, I will just close by acknowledging all of the fantastic and critical people that have been involved on these projects. So my wonderful advisor, Weizhe Hong, who I just couldn't ask for a better mentor over these years. Shan Huang, who was a postdoc in the lab and is a great friend and was my co-author on these projects. Could not have done any of this without her. And other people in the lab, Tara, Emily, and Patrick, as well as fantastic undergraduates, June and Leticia. And I also want to just quickly mention the several people at UCLA who have supported this work either through technical support, Paimon and Ping Ping and Daniel, or intellectual support. My committee members, Alcino, Satiris, and Dean have also contributed greatly to the different kinds of directions that this body of work has taken. So yeah, thank you so much, everybody, for paying attention and listening. And I guess we have a little bit of time for some questions, I think, right? Maybe 10 minutes or so. So I think that that's pretty good. Yeah, thank you so much.
Great. Thanks for the great talk. And there are quite a lot of questions in the chat box. So Felix, how should we proceed? I guess I can group some questions together so Lyle can answer them together. And I think if people, people can also ask questions themselves. So I guess one of the first questions that people ask is the nature of the barrier experiment that you did to separate the two animals. Can the animals see each other? Number one, can the animals smell each other? Is there any odor or pheromones that are being communicated? Could you comment a little bit on that?
Yeah, so we used opaque barriers, so the animals in the open arena we used opaque barriers, so the animals can't see each other. It's definitely still possible that pheromonal information or olfactory information generally may be diffused across that barrier, or there may be sounds the animals are making that we haven't measured. So if you recall from that graph that I showed, I don't know if it's worth going back to, but there is a sort of remaining piece of interbrain correlation after we introduce that barrier. And I mean, in some sense, understanding what other variables may be at play here is ongoing for us, sort of following up on this study. But I would speculate that some of those other things that are present in the environment potentially shared between animals does explain some of that remaining correlation between their signals. So that's the quick version. But if anybody wants to talk about that more, I'm happy to chat later.
Right. So another question is related to the nature of the self and opponent cells, or self and other cells, and so the specificity of the PFC versus other regions. So are the two different cell types belong to sort of the same circuit or different circuit, and how they may exist in other brain regions?
I see. So I mean, when we look at just the sort of spatial distribution of subject and opponent encoding cells in MPFC in our experiments, they are broadly intermixed. Like, we don't see any spatial segregation of those neurons really. And that's consistent with what other people have found broadly in this cortical region. Like, a lot of the neural responses tend to be sort of intermixed. We haven't yet looked into recording from different, like, molecularly defined subpopulations. So it's certainly possible that those variables are encoded preferentially, maybe in excitatory or different inhibitory subtypes. So that's something that would be cool to explore. In terms of other brain regions, we haven't recorded from very many. We have some preliminary data recording from motor cortex. And there does seem to be some encoding of, like, opponent behavior in motor cortex. But we do think that broadly the responses of these cells to opponent behavior is something that is probably somewhat unique to MPFC. And just to be clear, we don't know, I mean, we're sort of agnostic as to what really those signals are encoding, whether it's largely sensory or sort of some more abstract integrated variable. It's certainly possible that, for example, the opponent push encoding neurons in MPFC are driven to a large degree by, like, the somatosensory response. So there simply is information about what the other animal is doing, but we don't want to claim that this is necessarily, like, observational in nature. So I don't know if that was maybe the thrust of the question, but yeah, that's a brief answer.
Yeah, so there are a couple other questions regarding to the dominance behavior and the use of tube test in measuring dominance behavior. So maybe you could clarify a little bit of how you use tube test to measure dominance quantitatively. And also there's another question. So whether like over multiple sessions or days, whether a dominant animal remain dominant, or against different individuals or against the same individual?
Okay. Yeah, I see. I think I missed like a little part of the second question, but I think that I think I got most of it. So I think I can
Consistency across sessions or
across
days.
sections, right? So
Whether
yeah,
you remain dominant. Yeah.
right. So operationally, we defined dominance in this body of work as basically related to the animal's position in the tube. So traditionally, when people have used this assay, they let the animals push each other completely out of the tube, and they count each session like that as a trial, and then they find that dominant animals will shove their opponent completely out of the tube. We because we were recording from these animals and we didn't want to like go in and touch them all the time, we closed the ends of the tube. So it's more of a dynamic long form session where they can kind of go back and forth over the course of 10 to 15 minutes. So we used the average position of the animal as a measure of dominance behavior, or dominance relationship. The logic being that the position the animal occupies in the tube on average is sort of an integration of all of the individual push and retreat events, so that dominant animals will spend a lot more time on the other side of the tube. And so that's sort of the logic of that. And generally speaking, it is known that dominant animals will show a consistent behavior in the tube test. So over multiple days, they will routinely push out subordinate animals. In our recording sessions, we didn't record over multiple days. So in terms of the behavioral data that we actually quantified in this study, I can't exactly comment on whether it was stable, but we did habituate the animals for several days prior, and sort of just anecdotally, animals that tended to be very dominant in those habituation sessions were so in the tube test. So the answer to that question, I guess, is generally yes, but we didn't record over multiple sessions.
So another question is regarding the temporal dynamics during the tube assay, or over the course of that assay, whether there's any initial behavior that disappears at later stage of the assay, and whether that correlation could be a result. I mean, I'm trying to phrase the question. Could be a result of any learning process between the two animals, like during the test, the animal learn the status of the other animal and somehow become correlated?
Huh. That's an interesting question. That's a really interesting question. So behaviorally, all that we can see is that the overall like rate of behavior generally does go down a bit towards the very end of the session. It seems to be about constant for like 8 to 10 minutes. And then after that, the animals seem to get a little bit maybe bored. I don't really know, or they're, I don't know, probably they get bored or something. So, or maybe they figure out, what the dominance relationship is and they just don't interact quite as much. But that tends to happen at the very end of the assay. For the majority of it, I would say the first 80% or so, the behavior rate is fairly constant, and the sort of distribution of behaviors is also constant. So it's different between dominant and subordinate animals, of course. Dominant animals push more and retreat less, but that doesn't seem to change over the course of the session. That said, it might be the case that if you put two animals that have never seen each other before in the tube, there might be some sort of learning process that occurs. So I don't want to like rule out that possibility, but in our experiments, we saw relatively constant like behavior repertoire between the animals.
Right, and how about social hierarchy, like dominance relationships among a group of animals? If you, for example, image the behavior of a dominant animal over a subordinate animal, and then image the same animal against another animal who is even more dominant, what would you expect?
Yeah, that's the experiment I've wanted to do for the last like year, but I haven't done. I really don't know. We just haven't done that experiment. I mean, the reality is that a lot of these data were acquired over a very long period of time, and so we don't have very many examples of sessions where we've recorded from the exact same animal in multiple different environments. or if we did, it was separated by a lot of time, and so we can't make that comparison very accurately. But that's something that I think I really want to do. I mean, I think it's a really cool question. Does the sort of tuning of those cells and potentially the way that those dynamics interact with the other animals' neural activity, does that change fundamentally when you pit an animal against a dominant when it was previously dominant to another? I mean, I think that's super cool. I hope that we can dig into that.
Yeah, there's another clarification question regarding the first part of the talk where you talk about the increase of neural response to females in the male brain, but in the female brain, the encoding seems to be similar between males and females. That seems to give the impression that the female signals in the male brain is driving majority of the interaction. Is that the case, or that's more of a yeah, you can. That seems to give the impression that the encoding of females in the male brain drives the majority of the behavioral interaction between two animals.
The sex preference behavior,
or in general.
So yeah, I don't know. I guess it seems to, I mean, it seems to, from what we've done in those experiments, it seems to drive the relative motivation to engage and interact with another conspecific. And but I mean our manipulation studies using the activity dependent labeling showed that when we activated male encoding neurons, that actually did the opposite, right? That increased the preference towards male. So I think the view would be in animals where those female cells are more excitable, or for whatever reason they are more strongly driven by sensory cues, that does drive that behavioral bias, but that's not a fixed thing across all animals, or even across multiple days in the same animal. That's something that could potentially be dynamically shifted, such that if an animal then changes its behavioral state, that could be due to the representations in the MPSC and how those are translating sensory cues into changes in motivational state. That would be my guess.
I think I covered most of the questions. Felix, how are we doing on time, or should we?
I think we're at the end of time. So thank you so much to Lyle for giving a really stimulating talk. And I think just from the chat section, you realize there are so many questions, and we hope you stick around to answer them all before you leave Weizhe's lab. But I know that you have
I'd love to
ideas of maybe graduating before you answered all of these questions. So this was awesome, and thank you, Weizhe, for bringing Lyle to this moment. So I think
thank you so much, Lyle, for the amazing talk.
Yeah, thanks so much for the honor. And I'll just add at the end, if anybody has questions that I didn't have the opportunity to answer, please just email me or whatever, because I love to talk. So just tell me. I'm happy to chat about it. Awesome. Well, thank you so much, everybody.
Okay, thank you, everybody, and enjoy the rest of the week. And please come back for the next one seminars next Tuesday at noon.